Hello, it's me, Teddy Mo, the PhD himbo, or whatever Adele said, here to host the final installment of the SCoBIRC Cirque web series, or at least for summer of 2024. This episode, we're going to focus on the future of research and what a PhD student has to say about the research, what a PhD candidate is doing to actually bring about these experiments, and then a postdoc to see how she got here and how she plans to keep pushing forward in science. I'm super excited for this installment.
Hi, I'm Lydia Flowers. I'm from Louisville, Kentucky, and I'm majoring in kinesiology, exercise science.
Hi, my name is Alika Mualim. I'm a neuroscience PhD student going into my third year. I'm from Wayne, New Jersey.
My name is Lauren Hill. I am an agricultural medical biotechnology major, and I will be a senior in the fall. All right, so you're a student at UK. How did you get here?
So I am from New Jersey, and I did my bachelor's and master's. I did my bachelor's in biology at William Patterson University in New Jersey, and my master's also in biotech, also from the same university. And I decided to continue my studies at the same university because my PI there, she was a great mentor, and I did research in traumatic brain injury and looking at a specific potassium channel after brain injury and how that's affected. And so her passion really sparked interest in for me, like to be interested in research. And so when I wanted to continue doing research, she suggested or recommended that I come here because she did her PhD at the University of Kentucky in the biology department. So that's how I came to this university. I first started in the bio department, but then I realized that I still really enjoy doing traumatic brain injury research. So then that's where I transferred to the neuroscience department.
Very nice.
Yeah.
So what is your why? Like, why would you choose science?
So I was really interested in science at an early age. I was born with achondroplasia, which is a deformity or a condition that affects the limbs. And so I had to have a lot of limb lengthening surgeries growing up, and undergoing so many surgeries made me really interested in science and really passionate for learning how to treat different conditions and diseases, and being able to see how the physician who treated me, how he develops all these different therapies and adopts it from who he learned it from, was really, really cool. And I just wanted to first be a physician, but then I realized I really like research and drug discoveries, and so I wanted to continue doing research. Yeah. How about you?
Um, I just really want to fix the gap in like healthcare disparities, mainly for like Black and Brown people, because I've witnessed firsthand like the horror of that. So I've always wanted to close that gap.
For me, my why, I have a very strong passion for seeing people improve. I feel like that's what I'm gifted in, and I really enjoy doing it. And I enjoy medicine and figuring out like how the body works, and because we're just so like complex creatures. And so I feel that I would love to help people improve upon themselves and like be active participants in their own health through like exercise and nutrition. So
Oh, that's awesome.
So outside of lab, who are you?
I am an outgoing person, I would say. I like to get involved in the community. I'm part of the Iranian Student Association because I'm also Iranian. And so there's a lot of graduate students who just came from Iran and started their PhD here. So being part of that and helping them since I was born here, with the obstacle of immigrating to America and like trying to navigate everything, being away from their family. So I was the vice president. Now I'm the president of the association. So just organizing different like outings and parties, and we have like a lot of cultural events that we like to host. So that's a big part of my life, and just being part of that is great. And I like to hang out with the grad students and go out, explore Lexington since I'm not from here, and finding different like hidden gems, since it's like it's kind of spread out, but different than New Jersey for sure.
Yeah,
Awesome.
I can imagine.
Yeah.
So how would you describe your research as a poster pitch at a conference?
So, I look at cerebral hypometabolism following a traumatic brain injury. And so, when you have a physical impact to the head, you have these cascade of events that occur, and this leads to mitochondrial dysfunction, and which leads into this ion imbalance and calcium disruption, or a big calcium influx that goes into the cell. And this further leads to cell damage, cell death, and chronic neuroinflammation. And chronic neuroinflammation can lead to this accumulation of amyloid beta plaques, which is a hallmark of Alzheimer's disease. And so you have this connection between TBI and Alzheimer's. And we hypothesize that TBI can accelerate Alzheimer's disease pathology. And so we're looking at this in an Alzheimer's relevant mouse model. And so seeing how the metabolism can be impaired after a brain injury in these animals, looking at different techniques such as metabolomics, and seeing where labeled glucose is going through these pathways, as well as looking at the electron transport chain within the mitochondria, and seeing how the oxygen consumption is affected. And so seeing all of that, and also neuroinflammation. And then furthermore, my project is looking at this beta hydroxybutyrate, which is a ketone body, and it's produced in the by the liver, and it's elevated in times of starvation and in a ketogenic diet. So we can see how this compound can bypass the effects of cerebral hypometabolism, and it can be used as an alternative fuel source to treat TBI and Alzheimer's disease.
All right.
Okay, so how would you, I feel like you did a pretty good job, but how would you describe your research to someone who's not familiar with SCI or TBI?
So when a person has a brain injury, it can be from a car accident or a football injury, this can cause different cellular events to happen in the brain, and this can lead to inflammation and cell death, which further leads to Alzheimer's disease, or this accumulation of these protein aggregates called amyloid beta. Mm-hmm. And so we want to see how how this also is affecting mitochondrial dysfunction. Since we know after a brain injury, you can have mitochondria, which we know are the powerhouse of the cell. They become damaged, and they can't take up glucose, which is the fuel that we use, our brains use for energy. And so to see how beta hydroxybutyrate, which is a compound that it's an alternative fuel that is used when the body burns energy or fat for energy. And so seeing how if you have damaged mitochondria, how can BHB bypass that and provide energy to neurons to reduce the neurons from becoming more damaged. And hopefully we can see a reduction of the acceleration of Alzheimer's disease pathology, and also the damage or further damage from a brain injury that someone might have.
Awesome. I got it that time.
Yeah.
So when translating science, what's one topic that you feel is the hardest to explain to someone who isn't in science?
Definitely the metabolic pathways. And for me, going through all these classes from high school to college to post-grad, I always thought, oh, I don't need to know glycolysis. Like, this is such a very intricate, complex pathway. I don't need to know it. But then I start grad school and they're like, here, you need to know it. And so learning that for me was very difficult. And then to be able to effectively communicate it, I think that's also very hard, because you need to know it. You need to know where these substrates are going, the enzymes that are breaking them down and converting it into another substrate, and then where it's going next, and then why is it upregulated in some places versus downregulated in another. So learning about that is very detailed and complex, and so I think that would be the most difficult one.
Yeah.
I'm gonna cry just thinking about it. Oh my gosh.
What's your research project focus on?
Oh, for this lab, I'm in Dr. Mayer and Dr. DuPont's lab, and so they're doing clinical research, looking at ICU patients, and because you're just laying there for so long, you have muscle atrophy, where they have muscle atrophy when they get out, and so just seeing how they recover over time, and just gathering as much data as we can, and so we follow them for 12 months. And just do like strength tests mostly, but we also measuring like optimism and resiliency, and then general like getting around, if they're like how their life has been impacted by like the muscle atrophy. So,
awesome.
Yeah, and then we'll also take muscle biopsies and like get to see it at a molecular level as well.
Wow. Oh, that sounds really cool.
Yeah.
I saw a biopsy. It was, yeah. I had to step away. I was like, oh gosh. Yeah.
Are you interested in going into medicine?
Yes,
Yeah.
definitely not surgery. I know that now. But yeah, eventually, family science.
It's great to be part of research and just figure out what you like and what you don't like at an early time before going in and applying for med school, because I think that's what really shifted my focus, because I wanted to go to med school and then I realized I really like research, so
yeah,
it's great to be part of it.
This program has been really great, and I think my lab particularly has been really great, just exposing me to so much to be like, oh, I don't want to do that, or like I really find that interesting. So yeah, it's been really nice.
Yeah, that's how I feel about my lab.
Yeah,
I'm in Dr. Patel's lab. We're basically looking at how spinal cord injury can be affected by like sepsis and the effects it has on the body and recovery rate and things like that. And we had to do perfusions. And I thought I wanted to go into medicine. And now I'm like, yeah, I don't think this is for me. I think I want to go into anesthesia. So I think I'll just probably do like general anesthesia. I wanted to work in surgical rooms, but no, it's not for me.
No.
Yeah.
Anesthesiology is great.
Yeah.
It's a great field, and when I shadowed a doctor, I saw them working behind like the drapes of the patient, like just giving them the medication, and it was very relaxed for them. Obviously, it's very complex because you have to know how much you're giving the patient, but once you know that, it's a very cool thing to do.
Yeah, it's very chill too.
Yeah.
Like I don't have to, like I have to do stuff, but it's not like life. It's life or death. Don't get me wrong, but it's like I'm not performing brain surgery or anything.
Yeah.
So
yes,
there's not the gore and blood and hammers sometimes.
Yeah.
All right. What is a scientific topic that you would like to further investigate in your lab?
So I'd like to look at how a drug can reduce the effects of TBI and Alzheimer's disease, and to see that going into the clinical phase would be really cool, because then it would make me feel like, oh, I'm actually making an impact and I'm making a difference. So that would be really cool to see if the drug that we're looking at is reducing the metabolic deficiencies that we're seeing.
Yeah, that would be really exciting.
Yeah, it would be.
Yeah.
So how do you overcome like failed experiments?
There's a lot of failed experiments in
Yeah.
science. A lot of troubleshooting, a lot of like taking a step back and saying, okay, I'm done for the day. I'll come back well rested and try again. There's a great support system here. I have a great lab that I can like fall back on and go to them for advice and support. So I can say, okay, this didn't work. What should I do now? And so having that guidance is really great in a field where you're always making mistakes and learning. And so also going back into the literature, seeing what others did differently.
What's your biggest goal as a researcher?
So I would like to bridge the gap between basic science and translational research, clinical research, and to see if what I'm doing actually follows through and goes to that point of, hey, like you had a TBI, here's some BHB, you'll get better. So I think that's my biggest goal, like drug discovery and figuring out what we can do to better help patients and mitigate their suffering after a TBI or after Alzheimer's disease, because there's so many people that are affected by it. And it would be great to have patients have a better life expectancy or life outcomes and be able to get back to what they were doing before they had a brain injury or if they are suffering from Alzheimer's disease.
So going long term, like where do you see yourself in 5 to 10 years?
So in 5 years, hopefully I'll be done with my PhD and then starting postdoc and having postdoc opportunities to further my research skills and abilities. And then in 10 years, hopefully having my own research lab where I can continue doing research and mentoring students and looking at neurotrauma and neurodegenerative diseases. How about you?
You know, wherever the wind takes me. I plan to go to PA school, but like I know not everything's set in stone. So I'm graduating next year. That's where I see myself.
That's one step at a time.
Yeah.
For me, I'm hoping to be in med school by then. I'm taking a gap year, so thinking about pursuing a master's in nutrition during that time. And so hopefully med school. Yeah.
Awesome.
Yeah.
So what made you want to pursue being a PI?
I think seeing how passionate my PI is, and also my former PI in William Patterson, I think that really resonated with me, and seeing how they mentor students and that reward that you get from training them and seeing them through grad life and also undergrad life or their careers, I think that really made an impact on me, and I would really like to do the same for up and coming scientists of the next generation.
If there was one thing you could say to someone suffering from SCI or TBI, what would you say about the research being done here?
I think that the research being done here is great. There's so much funding that's going into it. There's so many grants that we are applying for to be able to continue our research and make sure that it's as accurate as possible. And the patients who are suffering from a TBI or an SCI can be reassured or assured that we are getting to find therapies and treatments that can reduce the injury progression. And so I think this is a great institution for anyone who wants to do research because it's well funded, well established, and we are slowly getting to looking at clinical translational research. And we are. There's so many labs that are looking at human studies, human samples, and seeing the therapies that are working and actually getting to that point of treating patients from that basic science lab and then seeing it through.
Could you expand more on what made you decide between doctor and researcher?
Can I be honest?
Yes.
Or, okay. Be brutally honest? Okay. So I really was like hard set on becoming a doctor, a medical doctor. And so I took the MCAT and I did not do well. I scored really poorly. And so I wanted to retake it. And a lot of people that I've read on forums, they said they retook it and they did better and they moved on and they were able to apply. But then COVID hit and I saw all these physicians and how much they were suffering in the emergency room, in the hospitals, and really sacrificing their lives for patients. And seeing that kind of made me rethink, and also with the fact that it's a lot of work, like hard work memorizing, making sure that everything is perfect, because if you make a mistake, it's a patient's life. Whereas while I was doing research, if I made a mistake, it's the mouse and it's okay.
Yeah.
Like, not that it's okay, but it's not a human.
Yeah.
So it kind of made me able to make that decision of, okay, I really like research. I've been doing it for this many years, and let me continue and still be in that clinical field, like being in that clinical stage, but helping patients indirectly. So being able to do that behind the scenes type of thing. Yeah.
How obviously it has, but could you go into more how mentorship has affected your career path and your decisions that you made?
So I had a great PI in my undergrad, and that's why I continued with my master's with her, and she was great. I was able to go to her whenever I needed support or I wanted to change my mind, because when I was applying for med school, I had she was the one who wrote my letter of rec, and there were other professors who wrote them as well. But then I realized I didn't want to, and I was kind of afraid to go tell her because she did all that work for me. And so then when I did go, she was like, "Oh, that's fine. Like we can regroup, and you can do your PhD if you want and do research." So I think having a mentor that you can be open and honest with is amazing, and it's really important and helps with your career path, because if you don't have that, it kind of deters you from science. And I know a lot of students who did their PhDs and didn't have great mentors, and they don't like research at all. And it's mentorship really makes or breaks what you want to do. And if you don't have that person that's passionate about science, then you don't really, or passionate about anything, you don't become passionate about it. And I think also now having a great PI that I'm able to also go to whenever I need, and having that open door policy really, really strengthens your long-term decisions of what you want to do.
Yeah, that's awesome. Thank you.
Well, thank you for your time. Thank you for this interview.
Thank you for you and your times, and I appreciate you having me.
Yeah.
Thank you.
As a PhD student, I love hearing the stories of other PhD students here on campus because we come from such diverse backgrounds. My background in undergraduate research is way different than somebody's background that they did in maybe undergrad research and their master's. We all bring something new to the table, and that's why science is so beautiful, because you're able to bring something new from your own unique perspective. Now we will look at a technique that John Vincent does in Dr. Baxter's lab. It's pretty cool.
Hello, my name is Jonathan Vincent, and I'm a fifth-year MD PhD student at the University of Kentucky College of Medicine, and I've successfully passed my first and second year of medical school, taken the Step 1 exam, and now I'm currently a third-year graduate student in the Department of Neuroscience, training under Dr. Adam Baxter. And my dissertation work focuses on how interleukin-1 affects synapse function and recovery following a traumatic brain injury in mice. And so to do this, the technique that I'm primarily learning is extracellular field potential recordings. And so this is through slice electrophysiology. And so today we're going to talk a little bit about the technique and what my experiments kind of look like. And so to bring you to this board here, the extracellular field, this is an example of an extracellular field potential recording trace. And so when most people think of slice electrophysiology, they think of patch clamping, when you're actually taking an electrode and patching onto one specific neuron and evaluating that cell, that neuron's function. Well, what I do with the extracellular field potential recordings is a more gross understanding of synapse function. And so when you look at synapse function, you obviously have your presynaptic neuron and your postsynaptic neuron. And so when you inject a current through the stimulus electrode onto a mouse brain slice, you get an example field potential trace recording. And so to kind of break this down, at 0, so on the y-axis we have our response, and on the x-axis we just have time in milliseconds. And so the midline here of the plot is 0. And what happens is we have this stimulus artifact here. That's us injecting a current over the brain slice. And then you get this little dip here, and then this response here. And so when we go back to the model of the synapse, you have your presynaptic neuron, which correlates to this fiber volley, or your presynaptic input. And then you also have your postsynaptic neuron, which correlates to this excitatory postsynaptic response. And so you can actually plot your EPSP slope over your fiber volley amplitude, and you can get a basal synaptic strength curve. You also get information like if you plot your fiber volley, if you grade the stimulus, so you have like think of it like a chemical titration, but varying current intensities. So we go from 25 microamps to 600 microamps. And you would imagine theoretically that with an increase in current, you should get an increase in fiber volley response and an increase in the excitatory postsynaptic response as well. And so with what my experiments entail is kind of subjecting mice to either a traumatic brain injury or sham or uninjured procedure, and then carrying out this technique and evaluating synapse function. So now that we've gone over the example extracellular field potential trace, what's important is electrode placement in electrophysiology. And so the two specific regions of the mouse hippocampus, which is the commonly referred to as like the learning and memory center of the brain. There are subregions in this hippocampus, and I study specifically the dentate gyrus and the CA1 regions of the mouse hippocampus. And so, like I said, electrode placement is very important. And so the way this circuit works in the hippocampus is information comes in through the dentate gyrus, gets fed into the CA3, and then fed into the CA1, and then out to the cortex or wherever it may go. So when we evaluate synapse function in the dentate gyrus in the CA1, we actually stimulate the peripherent path fibers that come in from the entorhinal cortex. So in the mouse brain, sensory information kind of comes together in the entorhinal cortex and then feeds into the peripherent path, among other pathways, but for this specific pathway, the peripherent path goes into the dentate gyrus. So we stimulate the peripherent path, and then we record in the molecular layer of the dentate gyrus. The molecular layer is where the actual dendrites or the dendritic connections or the synapses are, and so that's why we record there. This purple here is the cell body layer, so all neurons have their cell body and then they have their axons and their dendrites, and so we don't want to stimulate in the cell body. That would be a different question. And that's why we stimulate in the dendritic region or the molecular layer of the dentate gyrus. So then, as the pathway, the hippocampal circuit continues, the perforant path turns into another pathway that goes to the CA3, and then the fibers from the CA3 to the CA1 are called the Schaefer collaterals. And so again, it's important that we stimulate the Schaefer collateral pathway from the CA3 to the CA1, and then we record in the dendritic layer of the CA1, which is the stratum radiatum. So now that we've talked about the example extracellular field potential recording trace, and we kind of understood now where we are stimulating in the hippocampus and recording from in the hippocampus in the subregions of the dentate gyrus and the CA1, now this is kind of just example data of what we get. And so, like I mentioned, my dissertation work is evaluating synapse function and recovery following a traumatic brain injury. And so here's just a hypothetical data output that you would get from our extracellular field potential recording trace. And so the first one here with uninjured in blue and injured in red, we have our presynaptic excitability trace or curve. And to get this, we have our fiber volley amplitude, and remember that is the correlation to the presynaptic input following a current injection. And so, and then the x-axis here is our stimulus intensity. So like I said, we do a titration of varying stimulus intensities from 25 microamps to 600 microamps. And then we plot basically just the fiber volley amplitude. And so hypothetically what you would see is in the uninjured group, you would have a higher presynaptic excitability than you would in the injured group. And this could be different. This is just an example. However, these are functional measures of the synapse, right? So we don't actually know at the molecular level what is causing these changes. You know, if you're talking about presynaptic excitability, you have to think about, you know, the number of synaptic vesicles that you have, the amount of neurotransmitter release, or is there any synapse loss so you're not exciting or you're exciting the same number of presynaptic neurons, and so you're not able to have you would have a reduced total input when you're looking for a postsynaptic response. And so that's what we'll go to next. We'll go to just the standard input-output curve, which you would traditionally find throughout the literature, and that's where you're plotting your excitatory postsynaptic potential response slope, not the amplitude, although sometimes in the literature you'll see the amplitude, but we specifically do the slope. And we plot that on the y-axis, and then again with the stimulus intensity, the varied stimulus intensity on the x-axis. And so again, in a injured versus uninjured mouse, you would see that you would expect to see potentially a decrease in your postsynaptic response following the injury. And so that actually brings us over to one of our more important results, and that would be when you plot the fiber volley, or you plot the EPSP slope over the fiber volley amplitude. And so these are actually, what's unique about this plot is it's actually two independent variables, because you're changing, the dependent variable is the stimulus intensity, right? And so basically you would have 12 points along each line, but both of these would be independent variables, because with a change in stimulus you would get a change in fiber volley, but also a change in the postsynaptic response, or the EPSP slope. And so we plot it like this because really what we're asking with this figure is we're asking, well, okay, with the stimulus, with the current stimulus intensity, what is the presynaptic input, and with that presynaptic input, what is the postsynaptic response you're getting? And so that kind of varies throughout the curve, and traditionally, I believe in rats, you get a more sigmoidal shape. Currently, what we're finding, and what some other collaborators are finding, is that in mice it seems to be a bit more linear. But let's just stay with the traditional sigmoidal type curve. And so this is essentially saying that in an injured mouse, the red line here, you would have with the same stimulus intensity, you would have a lower presynaptic input or lower fiber volley amplitude and a reduced postsynaptic response or EPSP slope. And so, this is the basal synaptic strength curve, and that's what's so great about electrophysiology is with one animal you can get so much information. All right, so now we're where the magic happens. We're with our Scientifica's electrophysiology rig, and essentially what we have here is we have our microscope, our camera, our stimulus electrode. That's actually what touches the slice, and then the current gets conducted through onto the synapses. And this is the recording electrode. This is what picks up the signal and then gets put onto the computer. And this is our recording chamber. So right now there's not a slice in there, but our slices are incubating, and we'll do that momentarily. But first, how does all this work? So over here, we have all this machinery and equipment. What does it actually do? And so really the two main pieces that we have, or I guess three main pieces we have here, are we have our digitizer, our amplifier, and our stimulus isolator. And so what's important about this is that when you have a slice in the recording chamber and you're trying to get a recording from it, the actual physiological response that is there is really small, so you can't detect it. And that's what this amplifier is for. And so the amplifier increases the signal from the synapse's response, and then so the computer is able to read it. And so you can think about it exactly like if you're playing an electric guitar. If you're playing an electric guitar and you strum the strings, it's really quiet until you plug in an amplifier. And that's exactly what you're doing. You're increasing the signal so that the computer is able to read it. And then this digitizer, what does that do? Well, I believe that the physiological response is more of like an analog signal. And so when able to translate this physiological response for the computer to be able to read it, we have to digitize the response. And so that's how the computer is able to read the signal, and then the amplifier is to amplify the signal. And then the stimulus isolator is actually what directly sends a current to our stimulus electrode, which then gets carried over the slice. So I think next I'm just going to put a slice in here and we're going to see what happens. Hopefully my slice prep worked today. And so great, we got a response, and you can see just like the example trace, here's the millivolt response on the y-axis, time scale on the x-axis in milliseconds. And here at 0 is baseline, because you shouldn't have a response without a stimulus. And so when we injected that 200 microamp current, you see the stimulus artifact here, and then you see the response in real time. You see this dip here, which is the fiber volley, and then you see a clear EPSP response, and then recovery back to baseline. And so we do this for our input-output curves to gather basal synaptic strength. We do this at 11 to 12 different intensities, and then for one slice in one region, and we switch to a new slice. And so we can get up to 6 slices per animal, and then we usually try to get 10 animals per group. So thank you all for your time and for listening. This is how we do slice electrophysiology in the Baxtetter lab, where we do our field potential recordings, measuring fiber volley presynaptic input and the excitatory postsynaptic response from the postsynapse side. And so thank you all, and if there's any questions, always feel free to reach out, and very grateful for the opportunity at the Spinal Cord and Brain Injury Research Center here under Dr. John Gensel. So thank you all.
One thing about being in such a specialized field of research, especially spinal cord injury or brain injury, or we as the Spinal Cord and Brain Injury Research Center, a lot of times you can get a little pigeonholed when you start doing your experiments and you just only know what you're doing, which is why I was so excited to have this opportunity to go and film different scientific techniques, because it's like I never would have seen that in any other sense, because I'm so used to my injury model and what I do for experimentation, which is why I'm so glad that we get to share this with you all and the general public, so you know what we're doing, especially with your tax money. Now we're going to talk to Fernanda, who is from Brazil, and talk about her journey to a postdoc and how she plans to see herself moving forward.
So my name is Fernanda França. I am from Porto Alegre in the south of Brazil. I did my undergrad in biomedical sciences in Federal University of Rio Grande do Sul. And then after that, I got my master's in biochemistry in the same university. And I also did my PhD in physiology there. And now I'm a postdoc at Dr. Genzel's lab in the University of Kentucky. So what brought me to science was that I've always been very curious as a kid. And I used to say that I either wanted to be a doctor or a detective or a writer. So I think that it all connected now because I feel like as a scientist, I'm all of that. And also, my cousin, she started this undergraduate course in biomedical sciences, and then I would ask her what the classes were like and what it was like to study that, and then she used to tell me all these cool things that she did. I thought, okay, I think I want to do that, and it was definitely the right choice. So my why is definitely to make an impact in people's lives. I know this is a big why and a big goal to have. And of course, when you think about science, you think about even the little discoveries, the little achievements that you do can like have an impact in the scientific community, and hopefully that will make an impact also in people's lives. So my why is definitely like I have to have a purpose in what I do, and the purpose is to hopefully help people in some way. So, of course, as a scientist in the spinal cord injury field, I see my research helping patients with spinal cord injury. So, maybe helping unveil some mechanism that happens during injury, or maybe even a therapeutic target that can be used to help improve these people's lives. So, I recently was awarded a grant to work on a project about using liposomal formulations of azithromycin in spinal cord injury. So, azithromycin has been shown to have immunomodulatory properties, so it can help shift macrophage phenotype towards a more regenerative phenotype. And using a liposomal formulation allows us to target specifically the macrophages, and then have hopefully have less side effects. Since azithromycin is an antibiotic, we have to be careful about the systemic effects of this drug, especially in the context of spinal cord injury, where a lot of patients receive azithromycin as a way of controlling infections. So this grant was funded by Wings for Life. That is a foundation that sponsors spinal cord injury basic, preclinical, and clinical research. So this grant is funded for 2 years, and it can be expanded for another year. So after this time, I really hope that I'm gonna be able to get a PI position. I've always dreamed of having my own lab. So hopefully with this time and with this grant, I'll be able to have enough experience and enough publications to land into a PI position. So during my PhD, I work with spinal cord injury in Brazil, and I've always read a lot of the papers published here by SCoBIRC. So I got in touch with Dr. Genzel. I sent him an email telling him that I was interested in working in his field, and he very promptly wanted to schedule a Zoom meeting so we could talk a little bit more about that. And I was very, very grateful when he offered me the position to come here. So coming here was, I think the biggest difference, like the biggest shock after coming here, is definitely how research is conducted and how like well the research is conducted here, and how much resources we have to do the research here. So it was a good shock. It was a good contrast when coming here and seeing all these very well-funded labs, all the equipments that they have here, and also all the community here in SCoBIRC helping each other. That was a very big difference that I noticed when I first got here. Culturally, I think Brazil is very exposed already to American culture, so we have a lot of American TV shows, American movies, and songs. So it wasn't so much of a shock, but of course, Brazil is a very warm country. People are really warm, and I think that is the biggest cultural difference from Brazil to America, is that not that the Americans are cold, but compared to Brazilians, that is, I think, one of the biggest shock. Other than that, being away from my family for the first time. It's hard, but thankfully we have the technology that allows us to like do video calls every day if we want to. And I also found a lot of Brazilians here at UK that helps us feel more home and more close to Brazil. So outside of the lab, I enjoy painting, I enjoy watching a lot of TV shows and spending time with my friends, uh, my American friends, my Brazilian friends that I found here, and all the people from other cultures that I found here. I like to have fun with my friends and explore the city. And also, now that it's summertime, I like to be by the pool, be by the sun with my friends. And I also like to go to the gym. I think it's something that sometimes I had to push myself, but I always feel better mentally and physically after.
Thank you for watching part 3, the final part to the SCoBIRC Cirque web series, at least for 2024. Hopefully we will see you again with more stories and peeling back the veil even more to show you what we have to offer here at SCoBIRC at the University of Kentucky. To stay updated, please follow us on all of our social medias and our website so you can stay up to date on the latest news of what SCoBIRC has to offer. Thank you once again, and I'm so excited to see you in the future.